Hey, everybody. Can you guys hear me? All right, cool. How are you guys hanging with the whole digital learning thing?
Good.
Good?
Not, but it's fine.
You haven't become a zombie yet. It's a zombie who has been made part of the undead by attending Zoom meetings. That was a joke. It's okay if you laugh. For those of you who are here, the first thing I want you to do is get into Moodle, so you might as well start progressing there. I'll tell everybody else to do that as well when they get here, but to move things along efficiently.
Yeah. I was calling to ask how to get it, but he just sent the link to our email like 2 seconds ago.
Here we go with this shithole.
Kitty, you look like you have somebody kidnapped and you're making a ransom video.
I don't know why it's so dark. The light is on.
It's because you have a really bright background.
Yeah.
So your camera is picking up the light background and you're now just a scary kidnapper silhouette, which is cool. But it's just a little scary. Dr. Judd, are you here? Where is Dr. Judd?
I said it, I can't get it.
Yeah, you're fine.
Okay. Well, it looks like we're still getting some people coming online, so we'll give it another minute.
I couldn't find live classes for nothing.
Dude, I can't find it at all.
It's in the Outlook email.
Yeah, like.
He sent it on 12/27.
Well, like I got nothing from.
Hey, Dr. Judd.
Hello. Sorry for the
On-
delay. Trouble with the link. It's all working now.
On breakout rooms, I have attempted to recover to pre-assigned rooms, which is supposedly how you're supposed to get it to go to the pre-assigned rooms. And it just puts Keaton in room 2, and that's all it does. Are you capable of assigning people to breakout rooms from your terminal?
No. As a co-host, I cannot assign, but I should be able to move between the rooms.
Okay. Then what, if you could help me out, I need to do a couple of things here at the beginning, but then can you guide the students through sharing their experimental designs while I set up the rooms again?
Yes.
Okay, all right. So I have a couple of bookkeeping things to do. In a failed effort to get breakout rooms set up that were permanent, I created them through the Zoom web portal. And unfortunately, that seems to not be working. So in the process, it also forced me to send out the invitations in a way that was not nearly as intuitive. So in the invitation, so some of you are having trouble logging on. So here is the, can everybody see my screen? Nod yes, okay. So if you scroll down, you'll get to, after all of the dates, you'll get to weekly. And under weekly, it has this long internet address. If you type that into your browser and then hit return, it will add your, will add this meeting to your calendar. And then all you have to do is go and find it in your calendar. And when you find it in your calendar, it will actually show you the link. So here's the calendar for today. And when I open that up, in theory, it appeared for just a sec. When you open that up, it will give you a link to join the Zoom meeting. If you don't want to add it to your calendar, the link right below that should take you to the Zoom meeting, and you can just join without adding it to your calendar. In normal invitations, those things appear as active links for whatever reason. Doing it through the web portal, it didn't do that. So hopefully that will fix that with time. Second thing, if you go to the Moodle page, so here on the Moodle page is our Moodle. I'm going to turn editing off so that it looks kind of like what it should look like when you're here. So we're here on August 31st. If you click on August 31st, there's a figure that you'll need to access later on today. If you go and look at September 2nd, you'll see that there are 3 things in here. One of the things that you should do is read the instructions up here. So there's a statistics assignment for Wednesday that's due at the beginning of the class on Wednesday, central limit theorem and basic statistics. You also need this R script in order to do that assignment. And then Young et al. Is a homework assignment that's actually due on Friday, but Dr. Judd will introduce it on Wednesday, which is why it's in the Wednesday materials. These things are in the Wednesday materials for me because these are things that are due on Wednesday. And so in the description, it tells you these things are due on Wednesday, and the Young et al. Is homework that will be due at the beginning of the class on Friday. So don't just look at the items inside of the folder, but also look at the description, because that will give you hints on what you're supposed to do with this stuff. We haven't really had a lot of stuff on Moodle yet, so I'll generally try and be fairly descriptive about what's in the folder and what you should be doing with it. Sometimes I'll put things that I don't want you to look at before class, but I want them to be there and available for you. And so I'll put things in the folder and give you instructions to not open it until you're asked to open it in class, because I want you to not see something until it's time to see it because of the activity that we have that day. Labs are this week. Tomorrow's lab is at 7:45. You will meet either in White Science 219 or White Science 224. In order to do in-class lab exercises, we're having to split you between 2 labs. And so for my class tomorrow, I will be sending you an email later on today, basically telling which group of people to go to which rooms. When you enter the room, there will be Post-it notes on the tables, and I want you to sit at the Post-it note that has your name on it. Once again, bring your mask because we're all going to be in the same room together, and wear it the entire time that you are in lab tomorrow, and sit at the place where I've assigned you to sit, and don't move the chairs around and things like that because I've placed them there to make sure that you're all appropriately distanced from one another. The other thing about lab that's going to be different is there are going to be activities that you're going to be doing that require you to work together as a group. And I'm going to have to be moving back and forth between the 2 rooms. So there's going to be a point where I'm going to assign one person in the class to be kind of a facilitator, a person who keeps track of things for the times when I'm moving back and forth between rooms to monitor what's going on in the 2 rooms. So it's a little different than what we've done in the past, but not too different. But it does require me to split time between rooms. So I need somebody to kind of be in charge while I'm in the other room helping the other groups. So expect an email from me later on this afternoon about what room to attend.
A similar thing will apply for those of you on Thursday.
Okay, yeah. So now that all the bookkeeping things are out of the way, do you have any questions about those bookkeeping things? Okay, do you have any questions over what we were doing last time? Okay, Dr. Judd, if you could take them through the different groups and talking about their experimental designs. You were asked to get together with your group over the weekend, come up with a name for your group, and also to think of what an experimental design might be to answer the question, is parental favoritism responsible for the coloration of the baby coots, this bright orange, bright red coloration? And while he's taking you through that, I'm going to set up the breakout rooms.
Okay, so normally what we would do is go around to each of the groups and put your experimental designs up on the board so we can all look at them and compare them and talk through them. Instead, what I'd like to do is kind of go around each of the groups. You should have had a designated speaker, or you can designate someone else this time if someone wants to volunteer. I'd like you to tell us your group name and then tell us about your experimental design. As you go through this, you know, there might be certain aspects of it that you feel pretty confident about, or certain aspects that you're sort of unsure about. That's fine. That's why we're having this discussion. We're going to kind of try to take the best of each of the concepts that you all come up with. So, group 1, you want to share with us your group name and remind everyone who you. Well, I guess we have that in the notepad here. Just share your group name, and then share experimental design. If anyone else in the group wants to help fill in some details after that initial summary, we can do that as well.
Okay, so our group name is the Octopi, and it is Joseph, Audrey, Cody, and Brandon. Our experimental design was to fence off a section of the coot's natural habitat, introduce 5 different coot families to this controlled environment, paint the coot chicks in 4 different color shades. So one will be the natural color of the coot, we'll just let it grow. The second will be a bright red color. The third will be a dull red, and the fourth will be all black. And so we're going to repeat that step across 4 different coot families. And then we'll have one family that's just all natural chicks to act as a control. And then we're going to take the weights of each of the chicks from all 5 families and compare the weights to the controlled family to see if there's any outliers between like different colors of the coots. So we're testing to see whether brighter reds or duller reds or all black are favored more by the mother.
All right. Nice. Is that a good summary, or does anyone else from the group have anything to add? How about group 2?
Hi. We named the group B, like the bees live in the wild. I am Paula. The other members are Grant, Riley, Alex, and Keaton. So our experiment, we have controlled enclosure with multiple group of coots living separately. And the environment will be designed to be closest to their natural environment. So we have bushes, pond, lake. We don't have roof for like natural light and rain and wind. We have cameras hidden in the brushes around the cage to keep track of their behaviors. And then we rank babies with a vibrant level of their feather, and we tag them to different group. But like so like the most vibrant is gonna be group 1, and then the least is gonna be whatever number, depends on the chicks we have. We're gonna collect the weight of different chicks. I think we expect the chicks in one group to have the similar weight. We also need information to see how, like, the metabolism of a chick to kind of indicate the study factor, because the weight is kind of irrelevant without metabolism and other factors we don't know yet. So we also, because of that, we have to study the behavior when they are hungry and when they are full. And the other data we will have to collect is the survival rate of each group. We also want to see which group color have the most attention from the mom, which have less, and which have the least.
So Paula, let me clarify. So you're going to divide them up by vibrancy and then apportion them to different females. Is that what I understood at the beginning?
No, I'm sorry. It was really confusing. So we have the natural mom, and then they're like kids living in different groups. So the mom has their like her kids, and
Okay.
we just rank them. Like we rank the kids
Gotcha.
in different.
Okay, I understand now.
Yeah.
And what was your group name?
B, like BEE.
The insect.
The B team.
Got you.
Excellent. All right. And then group 3? Do we have a speaker for group 3?
Savannah.
Yes, that's me. Sorry. We are doing the Lions. That's our group name.
Lions, great.
Okay. So for our experiment, we decided to design decoy cute babies, and we're gonna make some of them dull and then some of them brighter than the other ones, in efforts to see if maybe the parents favor the brighter ones. So we're going to make a controlled environment and use observational tools. And then we're gonna make the babies. We're going to introduce the mother to the model children by creating eggs that they can hatch out of, and then let them bond. And then we are going to, so our control group is going to have all the same dull color, and then our variable groups are going to have the like one bright baby. And then we're going to observe over time and see if the mother is spending time with which babies. Maybe she's spending more time with the brighter ones, we don't know. We're going to observe the amount of food that's given, the amount of protection that the mother gives, and then what happens when they're introduced to threat factors. So some more questions, like we also have maybe some more questions. If the mother does favor the brighter babies, we could see like what trends they have, and then what happens if maybe the brighter babies, if the color does not affect how much time the mother gives them.
Thanks, Savannah. Can I ask a couple of questions of you and the
Sure.
group? In your variable, your treatment group, did you say that you would have one bright baby and many dull? Or would it just be the one?
It would be one bright baby and then other dull ones.
Okay, gotcha.
Right.
And then you were interested in this idea of favoring the red one or the bright one. Can you talk or remind me a little? I think you started to say this, but can you tell me a little bit about what you mean by favor, or how you would measure favor?
Sure. So maybe the mother is spending more time with the brighter baby. Maybe she's giving that baby more food. Maybe she's protecting it more whenever there are threat factors involved.
And can you tell me a little about these model babies that you all were imagining? I only ask because I can, if I doing this when I was a student, I would have been thinking about like, a little wooden cutout or something, but I wonder what you all were thinking of.
I think since we're unlimited with our price range, we're going to try to make them as realistic as possible.
All right, so we'll have to get a collaboration with the physics department upstairs or something, right? Or
Right,
engineering.
sort of like robots.
Yeah,
I was
that's
still
what I
hoping
figured
for
you meant.
robotic babies.
Yep.
That's what I figured you meant. Great. All right. Anybody else have any questions for this group? Paul? If not, we can do group 4.
Yeah, the fact that they're robotic babies solves what my questions were. So yeah. Who's speaking for group 4?
Hey, so I'm speaking for group 4. We're the dingoes. I just came up with that. We're group 4 dingoes.
Is this Ryan?
Yeah, that's me, Ryan. And all right, so what we're going to do is what we were thinking was we're going to have 3 separate coot families in the same like size of environment, controlled, in a little space. We can put cameras everywhere so we can observe it, And then what we're going to do is have one group with like when there's nothing wrong, where it's just the right amount of food and like everything's all good. And then see how the mother reacts to the different. I forgot to mention. We're gonna have 4 coots. We're gonna have 2 brighter and then 2 duller looking coots for each group. So 4 babies for each group. And then one group is just gonna be all good with all types of food and everything they need. And then the next group is gonna have the right amount of food, but also with the predator involved in that to see who the mother favors to protect. The third group, we're going to have a lack of food to see who the mother decides to give all the food to, and then just observe from there.
I assume that's going to be a non-lethal predator.
Or so it's going to be like a decoy. We're going to like make a little paper mache like water snake or something.
So this is a question that I have that applies to a number of the different groups. So the hypothesis here is the hypothesis of parental favoritism. And so the idea is that a parent has a bunch of chicks and the parent chooses who within that brood gets the food. And so that's one hypothesis. And the idea is that there's variation among individuals in a given brood. But what if it isn't that you'll just feed the brightest one, regardless of how bright it is, as long as it's brighter than the other ones, versus it's a more binary thing. If you're red, you get food, and if you're black, you don't get food. So this is open to Ryan or anyone else who's presented so far. How would you go about separating those two ideas? Is it really the parents comparing among individuals in their brood, or is it just see red chick, give it food?
Well, I'm sure each mother has certain biases that, you know, I think it depends on the coot probably. One coot might like red, you know, one might like yellow, one might like them all black, who knows?
Anybody else have a comment?
I think with the robot baby example, having a control group of entirely black chicks and entirely red chicks, if the trends are different in each of those, that would tell us if it's a specific color that changes it.
Did the octopi group have a comment on that? Or is that where you were, Cameron? No, that was group 3, yeah. The octopi group had an experimental design that might start to get at that. Do you guys have any comments?
I think that because we have, in each of our groups, we have the natural, the brightest red, the duller, and the black, that we can gauge, we can make a scale from 1 to 4 of which of those are given the favoritism towards. And then we have the control group of the all natural, so we can compare the two that way. If that answers your question.
Mm-hmm.
Yeah, that's good.
I'm just asking questions to get you guys thinking about the complexities involved. And so we have group 5 next. Is that right?
Okay, so our group name is Saitama. And for our experiment, we were
Wait, could you spell that?
I think it's Saitama.
Okay, what does that mean?
Angelina.
One Punch Man, bro.
Yeah, One Punch Man.
That means nothing to me. Okay.
It's an anime.
All right. Go. Oh, anime. Okay, gotcha.
So for our experiment, we were just going to focus more like on the food part of it and take like in a controlled environment. So like whether it's like fenced off or something, have one like completely black chick, one that's more like dull, not like completely black, but has some color, and then a very vibrant one. And for like several days at like the same time of day, we were going to put a scale in there with a certain amount of food on it, and then just measure how much the mother coot like takes off to give to each chick, and then like look at how much she gives to each one and which one's the most.
Are you thinking you guys need cameras to watch how she apportions the food on the scale?
Yes, we would.
And so it sounds like you're just using the natural variation within broods to set up your distinctions between bright and dull chicks.
Yeah.
Okay. All right, room 6.
Okay. Our group name is Ravenclaw. And so our experiment, how we would set it up is we do 1 month of outside wilderness observations of 3 different nests in their natural habitat. So we wouldn't build any sort of arrangement or anything. We'd measure the lifespan and weight of baby coots after a month. And then we'd also do not backup, but reoccurring checkups every 2 months or every 2 weeks after the initial month. We'd see who's the alpha and who's the runt of the pack of coots based on overall attention that the mother gives. Set up game cameras at night to see how the mother interacts with the baby coots, and assign a bird each number, and then detail each one of its individual physical qualities. So instead of measuring the brightness, see what is different about each baby coot that the mother would favor that one over another. The rate of success would be determined by who gets the most attention and who's heaviest at the end of the month. We would tag the baby coots at the beginning of the month to keep track of them through the 3 different nests, and then do the 2-week checkup for the following 6 months. And so the equipment we would need would be game cameras, scales, tranquilizers if needed, tags, and a tent and food for yourself.
Interesting that you include in your supply list supplies to keep the humans alive. It's something that you should never overlook as you're thinking about doing things.
I like how you guys were thinking about what else the mother might favor, and we'll talk about how you're thinking about doing that. But I like that you guys were thinking about, well, what if she's favoring something else, and we're detecting favoritism, but maybe not from the colors. But we'll talk about how you guys are thinking about that in a little bit.
Yeah, so it's always good to keep multiple alternatives in mind as you're thinking through these things. So this is open to anybody. As I was taking notes on each group's experiments, what are some commonalities that you saw from group to group in all of these experimental designs?
I think that a lot of people really just focused on like the color being like what the mother would favor instead of anything else besides the last group.
Okay, well that's kind of driven by the hypothesis. So you would expect that to be prevalent, I think. What else? Commonalities.
A lot of the groups were weighing the baby coots at the end of the experiment to base who was favored the most.
Okay, so weight was a common response variable. Okay.
I think almost all of us decided to have a controlled environment instead of just observing them in their natural environment.
Mm-hmm. Anyone else? Dr. Judd, do you have any questions?
I was going to ask them a slightly different question. If any of them saw things that another group decided to measure that made them think, oh, that's really interesting. That'd be, we didn't think of that, and that you keyed in on.
Last time, oh, go ahead.
Answering Dr. Judd's question that he asked, originally my group was going to eliminate the predators in our environment because we wanted to make the color and the favoritism of the mother feeding her young the most direct that we could see. But other groups saying that they're going to create a false predator to see how she favors in the protection sort of sense rather than the feeding was very interesting.
Yeah, I agree. That was one of the things I noticed as well. And the reason that sort of piqued my interest is because when we were making observations in the, when we were looking at the original image, we were mostly talking about food because of the mother holding some vegetation, and we talked about whether maybe she might be feeding them that. And in fact, that's where we're going to go, but that was one way of manipulating or investigating favoritism that I hadn't heard last time we did this last year.
Also, most of your experiments seem like they're on the mensurative side. A mensurative experiment is where you have natural variation, and you use that natural variation to kind of set up your experimental groups. And the ones of you who were going to be doing some sort of manipulation were either moving chicks around so that you had groups of dull chicks and brighter chicks, or you were going to create robotic chicks because you have an unlimited budget in order to do this. None of you suggested actually altering the colors of the chicks themselves. Why was that? Unless I missed something somewhere.
Maybe octopi? Did you guys? Yeah, here comes
Yeah,
Joseph.
we did.
Oh, I
We
missed
met.
it because I was putting people in groups. I wasn't paying as close attention.
Yeah,
Sorry.
no problem.
Okay, so you were, and how were you going to do that?
Yeah,
Remind me.
remind us. I was hoping.
We were planning on just painting the chicks. So we'd said that we were gonna have one that was, we're gonna have 5 different groups of coot families, and so we're gonna have one, and so 4 different chicks in the group. So one that's a natural color, one that's a bright red paint, one that's a dull red, one that's all black, and then we're gonna have a family of coots that's solely natural colored, so we can compare the weights of the natural colored coots to the weights of the families with brighter colors and duller colors.
Okay, cool. All right. Dr. Judd, do you have anything else?
The one other thing that I hadn't heard before that I thought was interesting was a group that would limit the amount of food the mother has to give to the offspring. I thought that was an interesting idea that I hadn't heard before.
Yeah, if you have a limiting resource, then you're more likely to have to make decisions about how you're going to apportion it. That's a good idea.
Thank you.
So, okay, cool. So we've heard a lot of ideas, and Joseph gave me the segue into the next thing that I want to do. So I'm going to share my screen with you, and I'm going to tell you what the people actually did. So can you guys see a spider on your screen?
Yeah.
My apologies. So here are the coots. I'm going to pull up my PowerPoint, and oh, except that I've got these stupid breakout rooms on my screen. So here's the adult, or here's the babies. And so a bunch of you were just going to use sort of means for saying, oh, this is a brighter chick, this is maybe a duller chick with fewer red feathers, et cetera, et cetera. But the researchers who actually did the study that this case study is based off of actually altered the color of the chicks. So they went into individual broods and they actually manipulated color. Now they tried dyeing the coots first. So they were trying to do what the first group was suggesting, which is make some of them brighter, then dye them black, etc. And they found that that removed the oil from the feathers of the chicks, and they ended up dying of the cold. So they ended up not doing that. But what they discovered is that the bright red portions of the feathers are basically out here at the tips. And so they found that they could pick up the chicks, and they could just give the chicks a little bit of a haircut, so to speak, a feather cut, that would trim off the red area and convert red chicks into black chicks. And that didn't cause them any problems. Seemingly, they didn't see any difference in survivorship based on that. And so what they did was they then erected a pretty complicated experimental design. So what they did was they created 2 sets of control chicks. One control was all orange and another control was all black. So if a female had a brood of chicks, they just picked them up, they pretended to cut them and then let them go. And then another brood of chicks, they would pick up the chicks and they would actually cut the red portions of the feathers off and then return them to the families. And they did all of this out in actual marshes where they were having to monitor the behavior of the chicks as they were being fed by the parents, and they would have to recapture the chicks periodically. And they measured many of the same variables that you guys are planning on measuring. But then in addition to these 2 different control chicks where everybody's either all orange or everybody's either all black, they then had experimental broods where there was a mixture of orange chicks and black chicks. And they randomly assigned which chick would get trimmed first. So in some cases, the first chick that was hatched would get trimmed. In other cases, the first chick would be sham trimmed, picked up and treated as if it was going to be trimmed, but not trimming anything off. So can somebody kind of explain to me why they might have these three different groups, a control group of all orange, a control group of all black versus a experimental group where there's a mixture of orange and black?
So they could see if the mom like abandons individual chicks, because she's gonna have to raise a family. If it's all, even if it's all black, it's like in her nature to raise the chicks. So if it's more likely for her to abandon one individual one than her whole family, I would think.
Okay, does somebody have another idea?
It kind of levels the playing field of all the chicks whenever they're all the same color. So it shows if she were to favor one black chick in the all black chick group, if it was really based on the color or if it was an external factor for why she was favoring them.
Okay, anyone else have thoughts?
Letting us see the differences from the control group to the experimental group for each color would also be very helpful.
How so? Explain that.
Like if the black control group, the mother treats them one way, and the orange control group, she treats them one way, but she treats that same color differently in the experimental brood. That'll tell us about the intermixing of different colors and kind of favoritism based on color.
So getting back to my question that I asked a little while ago, the idea of favoritism is that you have a group of offspring and you're picking and choosing among them which ones are going to get the most attention. If you have a group that are all orange, do you have that ability to then decide which one gets the favored treatment? Same thing with all blacks. If they're all black, there's no basis for favoritism because presumably all of the chicks look exactly the same. Versus if there's a simple rule, feed the red ones, don't feed the black ones. If there's a simple rule that is feed the red or orange ones and don't feed the black ones, what would happen in the control groups? Well, actually, don't answer that. So this is what they did. So we have a control group of all orange and a control group of all black. We have a set of experimental broods that are black and orange. So on the Moodle site, I have given you a figure from the paper. And the figure from the paper shows the things that they measured. They measured feeding rate, how many feedings per chick per minute, the relative growth rate, and how many of them survived, what proportion of the brood survived. And what we filled in for you is a box and whisker plot. Actually, we didn't fill it in, but it's filled in the paper that shows the orange chicks in the control broods. And our task for you in your groups is to figure out what the data should look like for black controls, orange chicks, and black chicks in the experimental broods, keeping in mind this is what they did. Control all orange, control all black, and experimental groups. So what I want you to do is I want you to go to the Moodle site. There's a PDF of this image in there. Download it onto your iPads. Use your Apple Pencil to basically draw in what the data should look like if it truly is parental favoritism, where the parent is actually comparing one offspring to another in their brood and then partitioning parental care to the orange chicks, preferentially to the black chicks, versus just a simple rule, feed the orange ones, don't feed the black ones. And what I want you to do is do that individually. I'm going to split you out into your breakout rooms. Do it individually, and then when each of you is finished, compare notes on what you found and be ready when we all come back together to basically kind of share what you all agreed upon. Are we clear on what the instructions are? Okay, I have put you in your groups. I have given your groups the names. I think I might have spelled dingoes wrong, but forgive me for that. And Dr. Judd and I will be moving from room to room, like we always do. All right, we'll see you back in about 15 minutes. So work on it by yourself, then compare with one another.
Paul, once they go, we'll check a question with you.
I already assigned you to a room, Nathan. Nathan, you have a question?
Sorry, spacebar. I see that. I think we're okay. Never mind. I've got the answer.
Tessa and Ryan, go to your rooms. Hey, Tessa, you're still here. Nathan, stay with me for just a minute.
Yep.
Yeah, so I still can't figure the breakout room thing. I don't know why it's not working, but I followed all the instructions that are online for it. But anyway, this will hold for this period.
Yeah. I don't know why I was having trouble moving between breakout rooms last time, but I'm going to try a slightly different approach. The instructions on Zoom say I can do it, so I don't know why I'm having trouble. If I can't do it at the end of this, I'm going to just call them.
I just assigned you to Saitama for now, so go there and I can see where everybody is, so I'll just try and avoid rooms that you're already in.
Okay. Got it.
All right.
Okay, guys, I have a question. On the very first graph, so on the side it starts at 0 and goes up to 0.8. So is the higher you go, the more the chicks are being fed, or the higher the amount of feedings? I'm very confused on the first one. If somebody could help explain it to me.
Pay attention to the label on the y-axis. It's number of feedings per chick per minute.
Okay, so if 0.2 feeds per chick per minute is a lot less than 0.8 feeds per chick per minute. Okay.
Yep.
Thank you.
And on all of those, they're arranged such that the higher the number, the more parental care presumably is happening.
Maybe just screenshot it and then add it to the notes or whatever. That's what I did.
To the notes?
Yeah, like, do you have OneNote?
Not at all.
Download it because it works really well.
Right.
And then you just add it to notes and you can draw on it.
You don't have time though. You can just mark up on your photos and you can draw.
Yeah, next time, like, you do it.
yeah, here at the beginning, we're going to be figuring out how we navigate this digital world that we're in.
There's definitely a lot of changes.
Yes, yes. So you can see that we're struggling with it ourselves. So.
I suck with electronics.
What's that?
I suck with electronics.
But you got into the group, so. So you guys still working on your individual answers? Okay.
I think the only reason I didn't do that was because, like, if all she has is black chicks, like, you know, I feel like she's just gonna feed them.
Yeah, and that's what I was thinking
But yeah,
about. I'm like,
no, I get what you're saying though.
yeah.
But yeah.
Yeah, but for the experiment, I put the orange a lot higher than the blacks because we are going off the basis that the orange are going to be higher.
Yeah, me too.
Did anybody else get anything different than that?
I think so.
I just hopped in. What did you guys decide on?
We all pretty much said that in both the controls, we think that for the most part they should be equal, because like if she just has all the same color, like unless she like favors a different characteristic that it's not about brightness, they should pretty much be equal. But then both experimental, we all pretty much said we think the orange will be higher.
Yeah.
Okay. All right. Well, then just hang out while we check on the other rooms.
Okay.
While I'm gone, decide what would it have meant for the hypothesis had the black controls been the same as the black experimentals? Do that out of earshot.
Quite a bit more variance for those.
Yeah, I did something really similar. I was trying to draw on my Mac though, so it doesn't look too hot, but the idea is the same.
Yeah, I got the same thing too. On the controls, I kind of kept them at like, kind of competing with each other, but then for the experimental, I have them at the two, extremes.
What would it mean for the hypothesis if the black controls were low, like you're putting the black experimentals? Just ask that amongst yourself, and I'm gonna go check on another room.
So I think the black controls, like the hypothesis for that.
Are you guys done?
Yeah.
Yeah.
Oh my gosh, you're done. What did you guys find?
So we all kind of had the same idea that with the control groups, we did the same little graphics and everything would be the same. But then with the experimental groups, the black chicks would have like lower on the graphs.
Okay, so you're basically making the distinction that when there's variation in the brood, that's when you have the opportunity to express favoritism, and when there's no variation in the breed, there's no opportunity for favoritism.
Yes.
Okay. If you had drawn it the way such that the black controls were lower than the orange controls, what would that mean for the hypothesis of favoritism? I'm going to go to another room, discuss this amongst yourselves after I'm gone.
I think the favoritism, or that would kind of mean that you're expecting no favoritism, or
I think when I was with you before, you guys weren't done yet. Is that right?
Correct.
Yeah. What did you guys find while I was gone?
all kind of decided that for the experimental, it'd be pretty similar to the controls.
Okay. How do you mean? Describe it column by column to me.
Column by column.
On the graph.
Okay. So like for like the feeding rate, the orange chicks would get fed as much as they did in the orange control. Same for the relative growth, because they're getting fed as much, they grow the same. And then for their proportional surviving, I think it'd be pretty similar as well.
And what about the black chicks?
Mine doesn't have anything on the black chicks, like it's blank.
Yeah, you're supposed to fill it in.
On both sides?
On both sides.
Okay.
So if you have the hypothesis of parental favoritism, it says things about a brood of all orange chicks versus a brood of all black chicks. It also says things about the differences between orange chicks and black chicks within a single brood.
Yeah, I think if preferential favoritism based on color is what's at play here, then the orange and the black control groups will get fed similarly to each other.
Okay.
In the experimental group, the orange chicks will be fed more than the black chicks, and the black chicks will suffer because of that.
And what would it mean if the black chicks in the control group were fed less than the orange controls?
They would. They wouldn't have as big as a growth rate, and maybe not all of them are surviving as much as the orange chicks.
But what would that mean for the hypothesis of parental favoritism? I guess I should have asked it that way.
It wouldn't be much of a comparison because it's all black in a control group, so it'd be just a color basis regardless of if there's a red or orange present.
Okay, yeah, that's why they ultimately had to have those two control groups, was so they could rule out just is there color present versus
like red chick feed red type of deal.
Yep, exactly. Okay, cool. Well, I think I'm going to end the rooms because I think everybody's about done. So we'll come all together.
Dumbest person that she's ever met in her entire life.
All right, can you guys hear me?
Oh. Yeah.
Yep.
Yes, sir.
Okay, cool. All right. So as I was bopping around from, well, do we have everybody? Let's see, who do we have here? We don't have everybody back yet.
Yeah, looks like we're a little short.
Let's wait to repopulate. Dr. Judd, were you able to move around?
I was not.
Stinker. All right.
I'm gonna go to Zoom support after class.
Okay. Well, after you're out of ecology?
Yep.
Okay. So you guys over the next 48 hours are going to maybe need technical support from me or Dr. Judd as you work on your stats homework. So don't be afraid to contact us for technical support because we can help you through the process of getting done what we're asking you to do for Wednesday. All right, looks like most, if not all of us are back. So one group share what they found. Doesn't matter which group.
I'll go. Our group pretty much said that all of the controls will probably be the same because, like, if they're all black, you're obviously just gonna like feed the black unless like you're favoring a different characteristic, I guess. But then in both of the other experimental groups, we think that the orange ones will be favored more.
Okay, someone from another group, what would it mean if they had observed that the black controls were getting fed less than the orange controls?
I think that would mean that she truly like favors the orange over the black, and then she doesn't want the black one, so she's not going to feed them, so they won't survive.
All right, I couldn't hear much of that. Could you speak up?
Sorry, we said
Oh,
that
there you go.
if the blacks like weren't getting fed at all, it would mean that she really only cared about the orange and did not want the blacks to survive. Even though there was no other option, she would just rather not have any than have some black.
Okay, so in an environment where there is no variation, she still just doesn't feed the black chicks, and so there isn't really favoritism. There's more this on-off sort of rule, which is feed a chick if it's orange and don't feed a chick if it's not. And so that's the reason that they had to have this control group, these two complicated control groups in their study. So let me share my screen again, and we will actually look at what they found. So this was their design, and so what I'm going to basically show you in the panel to the left or to the right over here is what they actually found. And so what they actually found was that for feeding rate, the orange and black controls were essentially not different, and in the experimental broods, the orange chicks were being fed at a higher rate from the black chicks. So I now have another question for you. Why would it go up in the orange chicks and go down in the black chicks? Why is the orange bar higher, but the black bar is also then lower?
Because there's like, you know, orange and black. It's not just all orange or all black. So if there's like, you know, a little less orange, then it's going to go up because.
So why couldn't the parents just work harder on the orange chicks while still provisioning the black chicks at the same level as the controls were?
It shows that she's intentionally neglecting them. She's not. She's favoring the orange.
So she's favoring the orange, and then as a result, what happens to the black chicks?
They lose touch.
Yeah, they
get left behind.
Why would that be a case out in nature?
There's limited resources.
Okay, who was that?
Me.
Oh, Dominic, yeah. So if
Yeah.
you have limited resources, there's only so much food to go around. If you're giving more food to the orange chicks, that by necessity means that the black chicks are going to be getting less because you have a finite amount of food that you can give to your chicks as a whole. And that's a good thing to remember, this idea of limiting resources. We're going to revisit that idea periodically. So when we look at relative growth rates, we see that the relative growth rates of the controls are almost exactly alike. But when we look at the experimental broods, the orange chicks are growing faster than the black chicks. And the end result of this is that there's no differences in proportion surviving in the orange and black controls, but the orange chicks are surviving slightly better, although not tons better, than the black chicks. So at the end of this, would you say that this would then be data that are consistent with parental favoritism, or should we reject parental favoritism as an idea?
It definitely
consistent
proves.
with.
Okay, it's consistent with parental favoritism. There were other data in the paper that we weren't going to have you work with, but there's a hatching order to chicks such that you have one chick, so they lay the eggs sequentially, and so the chicks hatch out sequentially. And the first chick that hatches out in each brood fares pretty well regardless of whether or not they're orange or black. But starting with the second chick, whether you're orange or black starts to have an effect on your survivorship, such that the survivorship of orange chicks doesn't really change much across the hatching sequence. But as the further along you are in the hatching sequence, if you're the fifth one that hatches out, your survivorship goes way down compared to if you're the first one hatched as a black chick. And the reason for that, presumably, is that for those couple of days when you're the only chick in the nest, there's nobody to compare you to, so you get all of the food. But as soon as you start getting compared to somebody, when the second chick comes along and it's red, then all of a sudden you're getting less food. And here, there's the figure caption for this figure. So what we've basically done is taken you through the experimental design process, the scientific method process. And for this class, I divided up a little more finely than maybe you've seen it divided up in the past. So I'm going to move so you can see the board a little more clearly. But it occurs in a series of steps. The first of these steps is observation, which you went through by observing the coots. The second step for me is formulating a question. In this case, a lot of your questions had to do with, why are the coots colored this way when they're babies and not colored this way when they're adults? The third stage of this is hypothesis formation. And we discussed this last time we met. So the question is, why are the coots colored this way? What are all of the possible reasons? You guys worked through that in your groups, and then we went through and kind of evaluated which of those hypotheses were worth kind of pursuing or not. Once you have a hypothesis, which is just a proposed answer to the question, you design an experiment. And this can be either a manipulative experiment, and theirs was manipulative. They actually altered the variable of interest, or you can also have a mensurative experiment where you use natural variation to set up your experimental groups. You then implement that experimental design. You get the data from that and analyze those data. And then based on that analysis, you come to a conclusion. And the thing that you're concluding about is whether or not your hypothesis is consistent with the data that you generated. Now, at the end of that, you may generate more questions, which then require new hypotheses, which then require new experiments. So this can be an iterative process as you slowly whittle away at the areas of a system that you don't know about. But this is the scientific method as we think about it here. Things that are not in most treatments of experimental, of scientific method rather, that you might not see is the question phase, the implementation of the experimental design, and the experimental design process actually itself. But this is not that far off from what what you'll see in a textbook treatment of this. And so we could have just given you this list last Friday and then moved on to something else. But one of the things that we like doing is creating learning environments where you can come to these things through a process of collaborative learning. And so this is kind of how we do things here. We'll also do plenty of lecture periodically, but just get used to kind of this format of doing class. There's a homework assignment in today's materials. It is about statistics. It requires that you download R to your computer, R and RStudio. If you go to the top of the Moodle site, let me see if I still have Moodle up. If you go to the top of the Moodle page, yeah, it is still up. There's a thing here called Biology 133 Lectures and Screencasts. These video sessions that we're having are being recorded. I'm uploading those to YouTube. If you click on that link, it will take you to the YouTube channel, the YouTube playlist that is for this class. And you'll see that there are two screencasts in there. One screencast is about downloading and installing R and RStudio on your computer. The other screencast is about how you open up the script and start working through the script. It takes you part of the way through the script that is on the Moodle site, but it doesn't take you very far through that script. Also, there are instructions in the homework assignment that also help you with this. If you're having trouble getting things installed on your computer, make an appointment with either Dr. Judd or I to get you through that process. It's a little bit of an involved homework, so start on it early rather than leaving it until 8 o'clock on Wednesday morning. Do you guys have any questions? All right, then I'll see you on Wednesday.
All right, thanks everyone.